My name is Ben Busby. I am here with my friend Ankita Das. We work for DNA Nexus and MIODx, respectively. And we're going to talk about computational immunology and specifically T cell receptor sequencing today. And I think obviously computational immunology is on the minds of so many of us as over the past year and a half, a lot of bioinformaticians and other scientists have really become sort of armchair computational immunologists, some in serious and really productive ways. And that has really been gratifying to me to see that and really to try to be involved in that. And now, we've been talking to people about sequencing viruses for years and years and why we should sequence viruses and discover novel viruses. And I think that has become clear to people. But also on the other side, the T cell receptor triad is so important. And so now we're seeing more and more companies doing that work. So, of course, MIODx is one of them, but there's lots of other companies, including Adaptive and several others, that are really interested in T cell sequencing. And why are they so interested? And honestly, the nature is, and I don't think it's an overstatement to say that if we understood the molecular nature of the entire triad, MHC, peptide, and the TCR, then we could tell people who got sequenced very, very early whether their T cell clones indicated bacterial infection, a viral infection, or cancer, and really sort of specific phenotypes within those groups. And so to me, that's really, if you will, kind of a holy grail of healthcare. And so hence the image. But there's challenges. There's a lot of bench immunologists who are learning to handle large, complex data sets. Some personal friends of mine, I would say, fall into that category. So we're working with them, and really we want to make this size data accessible to everyone else. And that's one of the reasons I'm proud to work for DNA Nexus, is because that's we're able to make this kind of data more accessible. I think on the flip side, up until, like I mentioned, about a year and a half ago, most computational biologists didn't know, actually, frankly, all that much about immunology. And I think this is something that's starting to change. And I think this may be, and this is a guess here, but this may be the decade of infectious disease research. I think we're seeing a lot more interest in not only COVID, but also other areas of metabolomics. And in addition to thinking about T cell receptors in the context of chronic disease. Finally, metadata is really complicated everywhere, and even more so in T cells and B cell receptors. So we'll talk about some approaches to that in just a minute. So, and here's one approach to the metadata. So this is iReceptor, and it's really made by the AIR community. So if you don't know what AIR is, check it out. Basically, they have suggestions for appropriate metadata for these types of sequencing studies, whether they be TCR or antibody. And if labs that are doing this kind of sequencing adhere to these metadata standards, they can get sort of an AIR stamp of approval when they submit to public databases. But what it really means is that these databases can be crossed with each other, and there's sufficient metadata to make this type of thing useful. And I think this is really important when we talk about repertoires, simply because of the complexity of repertoires. And Ankita is going to explain repertoire complexity for those of you who are not immunologists in just a few minutes. And then I will say that, this is, again, a personal interest of mine. And so I worked with the FDA to come up with a precision immunology apathon. And we looked at a number of these data sets, trying to think about COVID severity in a number of populations with T cell receptors. And I will say that there was some phenomenal data from Adaptive and many other places. And also, we're not going to talk about it here, but our collaborators at MIODx put in some really sort of neat machine learning work, and you can read about that on the FDA website. And again, we used the data was harmonized by iReceptor for that particular challenge. And I hope that there can be future challenges like that as we start thinking about having community responses to other infectious diseases. So that's really neat. And as I mentioned, I do all this under the auspices of a place called DNA Nexus. Really, our platform is it's aimed at everyone across sort of the biological data science spectrum, but really it enables bioinformaticians to be more powerful. And I think that's a really amazing thing. So I had a collaborator, and he said, "Well, I have a full-time bioinformatician to go on this project for a year. What do I need DNA Nexus for?" And I said, because then he can be on the project for a month and do 11 other projects. And I'm trying to make that real reality for him. So that should be exciting. And basically, yeah, I mean, for some sort of any sort of clinical diagnostics company, again, any test, any sequencer, we're friends with PacBio, we're friends with Nanopore. So we can, you can onboard any algorithms, or our professional services can onboard any algorithms, and we can make that work. So instead of rolling your own, certainly it's valuable to think about leveraging DNA Nexus, as our collaborators with MIODx at MIODx have. But then after you do these sort of basic secondary pipelines, I think really the exciting part and the part my group works on, mostly in the context of the UK Biobank, but in other realms too, is thinking about how to make Jupyter notebooks that work with it for a range of use cases in the biological data science space. And so working with Jupyter has been pretty fantastic for us. So with that said, I'm going to turn the floor over to my colleague and friend Ankita Das, who's going to tell you a whole bunch about basic computational immunology, how to think about sequencing T cells, what she thinks about with sequencing T cells, some applications, some really funky machine learning stuff. And I'm going to interject in the middle of her talk on purpose to explain something called SHAP, for explainable, a tool for explainable machine learning. And then she'll continue talk about some of this stuff. And then I'll come back and talk about visualization and really utility of these Jupyter notebooks at the very end. So with that, I'm going to mute myself and turn the floor over to Ankita.
Thank you, Ben. Hello, everyone. Good morning. And my name is Ankita. I'm representing MIODx. We are a genomics company in the Bay Area that focuses on using immune profiling data sets and building tools to enable biomarker discovery. Okay, so what I'll start doing is Ben has already introduced the importance of, or the work that's going on in the community and utility of looking at T-cell biology. However, I'll go a little bit into the background and show what is being done in the field and what we are working on. So what's shown on the left here is a cartoon that represents the diverse immune cells of the adaptive immune system. However, what you can see are these T and B cells standing out because they come in many flavors. And what's shown on the right is how are these T cells responding in the case of an infection and immunotherapy treatment in cancer. So at a very high level, what happens is when there is an infection and the T cells can sense it, they undergo this phenomena called clonal amplification, where a subset of the T cell repertoire, T cells will amplify and then respond to the virus and induce an immune response. The other thing that happens is if you have a tumor and you're doing an immunotherapy to treat for cancer, often the T cells are then brought to the tumor, and by assaying for the changes in the T cell clonality, one can actually understand the diagnosis or whether the treatment is working. Hence, looking at the T cells can in fact guide like diagnosing a disease as well as treatment. So again, I'll go a little bit more into what is it about the T cells in this slide. And so it's not, what's been studied is, of course, there are the T cells, and what influences these T cells to behave a certain way is genetics of an individual, the environment that they're in, certain lifestyle, as well as age. I'm bringing all this up is because at MIODx, we're not only building tools to look at the T cell genomics and the TCR repertoire, but also factors that influence the biology or the immune health of an individual. And what's shown in the bottom is the point that, and getting deeper into the role of T cells and the immune responses, where, as an example, if there's a virus infection, not only are these T cells getting amplified that can lead to a viral clearance, but they can also induce other types of, or engage other types of cells, such as the B cells, and lead to antibody priming and generation, making the point that really the T cells are at the center of and orchestrating the immune response, whether it's generation of antibodies or killing of the cells that have the virus infection. So at MIODx, this is why we're looking at the TCR, and I'll get into the TCR repertoire to understand immune health better. So what is it about the T cells that makes them so special? So these T cells have these receptors, the TCR, and they're highly diverse, and it ranges in the peripheral system and in the tissues, and it's in the range of over 10 million of them. And the way the diversity is generated during development is shown on the right. So what you see here is the germline DNA of the T and B cells, and I'm showing only focusing on the TCR T cells today. And you have these gene segments, the VDJ, and there's this very specific molecular recombination event, the VDJ site-specific recombination, which leads to the joining and selection of specific segments of the V, D, and J in the beta chain and the V and J in the alpha chain. And then once this recombination event has happened, the transcription occurs, and additional splicing leads to the final product that's shown here, the TCR beta mRNA. And these, you can actually use in the field, individuals can study the DNA or the RNA to understand these T cell receptor genes. However, you need a very sensitive method because there are so many copies or so many different variations of the TCR repertoire available. So what I'm showing you here is what are the different possibilities and how do you pick a method of choice? And this sets the stage for what I'm going to discuss about the work we're doing at MIODx to harness the repertoire to build signatures to understand immune health. So in this cartoon, what you're seeing, each dot is a unique T cell clone. And there are various methods. So you can use an RNA and a 5' erase chemistry, which lets you get the full length of the TCR, or a single cell TCR sequencing, which lets you get the alpha beta repertoire, or use a genomic template, which only lets you get the CDR3 region. Now, if you are interested in understanding the full diversity and identifying the low frequency clones, you're better off going with the 5' erase-based method. However, if you have a scenario of a disease wherein certain clones are amplified, so they're present at a higher frequency, if you now use a genomic DNA-based method or any of the methods, you will pick up the top clones. So if you are only interested in understanding top clonotypes, any method is good. However, if you want to get a broader and a deeper information about the repertoire, you probably want to stick with an RNA-based method. And single cell is very valuable if you want to understand the alpha beta pairing of T cells and also the transcription or the state of the cell of a single cell that you might be monitoring or putting back into a patient in a cell therapy. So, and the other things to keep in mind, if you look at the flow of how TCR data is used, is not only do you need to have a method that identifies TCRs, you want to have an analysis, like the bioinformatics needs to be scalable and easily accessible. So as Ben introduced, a lot of folks are immunologists and not computational immunologists, and some of them are not familiar with the biology, so you need to have robust tools. The third piece of this is, even if you have TCR data, how do you make sense of data? And this is an area where we're putting a lot of effort as a community as well as at MIODx. So how do you compare with other repertoires, bring in public data sets and draw insights? And I'll show some work we've been doing in this space as well. So some of the challenges, and Ben touched upon this a little bit, is that there are efforts in the community, such as the iReceptor, and of course, PubMed has publications with TCR data in it and other databases. However, they're all in individual domains that may or may not be accessible to a central. So if a researcher generates data, they have to go and access the servers where this is hosted. And even the analysis pipelines are not all in one place. So because of the challenge then is you really need a centralized architecture to really bring your data, bring all the metadata combined with other data and draw insights from it. And so in the next slide, what I'd like to present is what is it that we're doing with, this is why we partnered with DNA Nexus, because they have a scalable cloud-based architecture to seamlessly build bioinformatics pipelines, store and manage data in a secure environment. So here's what we have done. So a researcher can bring bulk TCR repertoire data like FASTQ files onto this cloud analysis tool and called the ClonoMap. And they can not only, by clicking a button, they can upload data with metadata and run analysis. And what you end up with are these visuals, which tell you what are the top clones or what are the clones and how they're changing over time. In addition, we have this other product, the Immune Insight, which lets you search in the environment, connect to the databases, staying within the DNA Nexus environment, bringing the data and combining and collecting data over time. This allows you to not only do experiments and analyze your own data at scale, but also combine data over time and build and look at time course analysis, which could be of importance to folks that are doing clinical trials and trying to understand how an individual's immune system is changing before and after treatment, and build biomarkers by taking a retrospective approach. So what I'm showing you now are two examples wherein we applied these tools to COVID-19, and this was touched upon as there is now an effort from the FDA as well as other groups to generate a lot of TCR data, various chemistries. So what we did is we wanted to use our tools and look at the role of T cells in COVID-19, and we took some public data sets. In fact, we searched the public data using the Immune Insight tool and then uploaded them into the DNA Nexus portal, along with the relevant metadata of the outcome, meaning folks that are healthy versus have severe outcome in COVID versus mild, and look to see how the T cell clones were changing. And in fact, what you see here in this graph is the CDR3 clonality. Of the left is of healthy individuals, and in the middle you have two individuals that had severe COVID, and we took the data from TCR over in a time course. And what's shown nicely is that compared to the healthy, where majority of the CDR3 clonotypes are present at a low frequency as expected, in the COVID, there are TCR clones that are found to be at a higher frequency in individuals with severe as well as mild COVID, suggesting that there is a T cell response going on here. And in fact, you can go back and compare individuals and see that there are shared CDR3s, meaning they might be pointing to one antigen-specific T cell. The other thing we found is we then looked at the V gene usage, and we saw some V genes higher at a higher frequency in COVID individuals, but what stood out to us was this one gene that's boxed in red. And in fact, that was high not only in COVID, but also in a healthy, and we wondered what that was. And again, because we have this Insight tool, we could search the V gene, and what came up was this public clone, which is found in in individuals with flu or a flu vaccine. So what I'm trying to say here is, using this tool, not only were we able to point to the potential role of T cells in the immune response of COVID-19, but also look at other diseases or other underlying factors that could be altering the outcome. So this kind of tool, this needs a lot more work, but it allows us to build hypothesis and do more discovery and come back and combine more data and see if your hypothesis holds true. So in the next example, we actually sequenced a limited set of rheumatoid arthritis individuals and healthy PBMCs, again, using a 5' raised chemistry. And here what we were asking is, are there any T cells, TCRs, alpha and beta, that are different between healthy versus RA? And in fact, we grouped the patients and we find that there are TCRs, I've listed some there, that are different between RA patients and healthy, pointing to some being either, they could be specific to antigens that are found in RA patients. So, and then we also asked, are there, on the right, what's shown is this time course analysis or shared clonotype analysis with the CDR3. And in fact, we see that their patients with RA have shared clones, and we can track them over time. So we're doing more research to understand what does this mean, but this is a way that we can start to define and pull out T cell genes of interest by essentially an automated pipeline that generates these visual outputs. Okay. So now I'm going to actually go back to, I started by saying that at MIODx, not only are we trying to understand or build tools that will help you find TCRs and relevant for your research, but also other factors such as age and genetics. So I'll show some work we've been doing, and I'll actually hand it over to Ben, because this part we have applied machine learning and built machine learning tools that helps to do more sophisticated analysis and draw associations between genomic data and other kinds of data.
So yeah, I'm going to talk just a little bit about explainable machine learning. Basically, when you leverage any particular machine learning tool, then you get a variety of features, particularly if you have phenotyping or even deep phenotyping of folks. You get features, and those can be phenotyped, or that can be immune repertoires. And so with explainable machine learning approaches, in this particular case, SHAP, basically what you can do is weight how much each feature contributes to your model. And really, we've done a fair amount of work with that. In terms of adverse drug responses, we gave 4 short webinars on it earlier this year. But I think, as you'll see in a moment, the MIODx team has done some really exciting work with this too. And so I will turn the floor back over to Ankita and let her describe some of those experiments.
Okay, thank you, Ben, for explaining, giving a quick introduction on SHAP and essentially how machine learning can help guide biomarker discovery from genomic and other phenotypic metadata. So what we've been doing at MIODx is we have this profiler and insight, which lets you analyze your own data, bulk repertoire. However, we are internally building a database taking healthy TCR data and metadata. When I say metadata, this means other types of information like age, sex, etc. And we've run it through our immune profiler, which helps us generate frequency of clonotypes and insights to bring in other tools. And then we're building our immune AI product, which is essentially a machine learning algorithm that we've applied to the healthy data and builds a signature. And what this signature does is it allows you to predict immune health. So just to give a little bit more in-depth on what that looks like. So essentially what the machine learning does is it goes and looks for features that are predictive of an individual's health and ranks the features. And what's shown are these SHAP plots here. As what that does is it helps to determine or let a researcher see visually how a feature in a TCR or any other data is important for predicting or the hypothesis that they are generating. So in this case, the way we applied that is using the machine learning tool, we define features of importance that are predictive of health in such that if you now overlay another individual's data on top of the baseline, it will help you see how these features are relative to the baseline, how it deviates from the baseline, and rank those features and tell you whether, if you want to focus on a set of TCRs to build a biomarker, which one should you be focusing on? So I'll show, and in the case of COVID, we actually took the COVID data and looked at it. So we have certain features that tell you that if you have more of that, it means you have a worse health outcome. And in fact, and I'm not, this is proprietary, in fact, when we applied that signature to COVID TCR data, we see that the individual with COVID had high of that feature, suggesting they are unhealthy. All right. So in this slide, I'll end with how we've applied this to rheumatoid arthritis now. And what you have on the left and the yellow line, trend line, shows the baseline, and the x-axis are these different features found by the machine learning algorithm. And the RA patient, obviously, you can see in the blue line is different. So it has a deviation from the healthy, meaning it is diseased. However, I want to point attention to this, the magenta or pink bars, because what that tells you, what you can see is if you look at the x-axis here in the deviation from healthy, the very first feature and the third feature, they both seem to have similar deviation from healthy. So if you had not applied machine learning, what you would think is, okay, both of these TCRs are potentially important, equally important, or similarly important in predicting or suggesting that these TCRs are important for RA. However, with the machine learning, as Ben explained, we're able to point and rank the features of what is most impactful from the signature. And with that, we see the top feature that's shown here, which is a TCR gene feature. And in fact, what we then did is we went back and looked in the literature to see if anyone's reported any of these features being reported. In fact, we found a paper where they had taken our individuals treated with the methotrexate and had responders and non-responders and collected PBMCs and did TCR. And I'm not showing you in detail, but we found that one of the top features that is being said to be impactful based on our machine learning, in fact, was most differential between responders and non-responders to the drug, suggesting that this approach with more data and more validation can be, in fact, be utilized to prioritize candidates for biomarkers for drug for patient stratification. So the last part of this, Ben has a lot to say after me because he's been doing work on this, is essentially looking at genetics and looking at how we can apply some of the immune profiling information along with population genomics to see how in different populations we can define biomarkers. So the approach here, and this is a research approach, is where you can take whole genome sequence data sets as well as race TCR data sets. Because there is full length coverage, you can actually impute back what you're trying to do is look for allelic differences in the TCR genes, because our machine learning has pointed us at a very granular level that there might be certain features within the TCR that can be pushing someone's health in a certain direction, and look at those variants and then associate them with disease outcomes and come up with risk scores and potentially build a TCR-based risk score for certain diseases. And in fact, the next slide, we were very excited to see that recently there is a study that has now taken TCR data, built a database, and looked with different methods, including full and 5' race, has found a way to novel alleles. They've, in fact, identified novel alleles in the TCRs, and there's some discussion on how this could be associated with disease. So this is a direction we're pursuing to further develop biomarker approaches at MIODx. And of course, we're going to do experimental validation and additional experiments and TCR analysis to really go after this. And then with that, I think I would like to turn it back to Ben for his additional work that his group has been working in this area.
Thanks, Ankita. Ankita does have a next step slide, but let's talk about that a little bit in the Q&A, and I'm sure some of you can think about that. And she touched on all of these points as well. So I think one thing that's been really sort of underlying a lot of the work that the MIODx group has done is the ease of use of Jupyter Lab and some very powerful compute that underlays the Jupyter Lab at DNA Nexus. In addition to the fact that it's actually possible to build web apps. So for specific clients, we can build web apps, or clients can build web apps for themselves or their respective clients. And I think that's been something that's been really useful. And I just want to show you a couple of graphics that have come actually not from web apps, but straight from the Jupyter notebooks that we've provisioned in collaboration with MIODx. And so, again, I think data exploration with Jupyter is very exciting. And if you're not a DNA Nexus client and you're not a MIODx client, and maybe you do something else and you just sort of have an armchair interest in computational immunology, check out the Jupyter notebooks. You can go and check out data carpentry and learn lots about Jupyter notebooks. The carpentries are fantastic for learning about lots of technical things, even if you're not completely submerged in computational immunology. So a nice thing about the DNA Nexus's particular implementation of Jupyter Lab is that really we make it very easy to use really sort of community-wide tools like Bioconductor, Bioconda, and Docker's there somewhat natively as well. So that's a nice thing. And then, of course, GitHub and cross-posting to GitHub. And really, we're working on an explicit community where folks will be able to build notebooks and then share them with other folks at DNA Nexus, both in the UKB project as well as in general. And I think that could be very useful for both research as well as other types of collaborations. And again, the web terminal is very flexible. That's really nice. I do all kinds of work. My team does all kinds of work on this, including all the way up to things like fairly complex image processing. So that's really quite nice. And we have a really easy to use Spark layer. So if you need to slurp really big things or move data from our Apollo platform, that's something that's relatively straightforward to do. But thinking about sort of just basic Jupyter functionality, we're really able to enable visualization. And I think that's really nice. So again, I talked about our platform sort of being focused on informaticians and biological data scientists. And this really allows them to spin up, get past the notebook, and then be able to create these visualizations of things like gene usage correlation, t-SNE, and then gene usage, of course, as well as thinking about CDR3 length. So I think that's really nice. And we're really able to port in software and make graphs like this on the fly. And I think that's really a big strength, simply because there just are so many use cases in computational biology and even computational immunology that we want to enable folks to tackle. So that's something that I think is very cool and definitely approachable. Finally, I think this is sort of the big deal for everyone here. If you are a computational immunologist, is thinking about clonotype tracking. And then again, with the machine learning stuff we talked about a bit ago, then we're able to think about which clonotypes can actually act as features. And so that I think is really sort of cool and on the cutting edge. And hopefully that was inspiring to you as you listen to it. And if you're interested in, maybe you're not a computational immunologist or your project is different, but if you want to talk about, using machine learning to work on some of these issues, please go ahead and reach out to DNA Nexus, and we'll get somebody, maybe me, on the horn with you to talk through some of these issues. And last, I want to talk about something that I am personally very, very excited about, and that is the idea of inferring some of these rearrangements and some of these repertoires from germline. And again, some of the reads are going to be informative, some of the reads are not going to be informative, but I think there's an active area of research, as Ankita really alluded to quite a bit, in terms of pulling these data from whole genome sequencing. And what that allows us to do is leverage massive scale data sets like the UK Biobank, exomic and soon whole genome sequencing data sets to look at some of these questions. Again, we have to do computational inference because we're using DNA, but it is possible to infer. But the other thing we can get from these biobanks are massive, massive phenotype data sets. And so if we can get things, like autoimmune information, severity information, that kind of thing, and mix it with these sorts of observations, I think that's going to be an extremely powerful tool, not just for infectious disease, but for chronic diseases as well. Now, one thing you may be wondering, you say, well, okay, but how is all this data stored? I mean, for the UK Biobank, you're talking about 500,000 people. That's absolutely massive. I'm talking about crossing on a number of variables. It just so happens that DNA Nexus has a platform called Apollo that is equipped to handle hundreds of thousands of individuals, both with genotype and really, really deep phenotyping, as well as imaging data, in fact. So if that's something you're interested in, please talk to us. If you're a UKB user, the UKB research platform will, in fact, enable that tool. And so that's something we look forward to integrating with data like this. And then on a personal research note, one thing I'm extremely enthusiastic about is thinking about getting Repertoire data, actually, as well as HLA data. And if you're interested in that side, check out the Dash hackathons run through Martin Mayers and the Be the Match Foundation. But anyway, getting this type of data into electronic medical records and into the hands of clinicians, because I really think that's going to make a difference for people. For example, cancer patients who rely on early detection to stay healthy. With that, I'd like to thank a whole bunch of people that really contributed a lot to this. Nick Hill, first and foremost, who did a lot of the technical Jupyter work. Mike, who did a lot of the machine learning work that we showed here. A whole bunch of other folks, thanks to iReceptor and the AIR community. If you are a computational immunologist, check out getting involved with the AIR community. It really is a phenomenal community in bioinformatics, and in my opinion, we need more of them. Thank you to Caroline and Brenton for putting this together. And with that, I think Ankita and I will take questions. So with that, there is a question from the audience, and this is certainly for Ankita. So I'll go ahead and ask, and then she can answer or defer. I'm sure she won't, as she wishes. But multiplex PCR-based NGS data can be heavily skewed. How does MIODx normalize that data?
Thanks, Ben. I'll answer the question. So first off, I think what you're referring to is using the data for the machine learning. That's what, because we've built this database. And what I want to say is we're not using multiplex PCR and we're using 5' erase data, which is ends up being less skewed, to be honest, but all TCR data is anyway a sub-repertoire, we're subsampling. So that's first part of the question. Second is, how are we normalizing it? So the machine learning has a way, the algorithm has a way to normalize the data by, and I'm not going to give away this in a much detail we can discuss offline, but there is a way that we normalize the data and make sure it's subsampled in a way that there is no bias in using the data for building the training set.
Thanks, Ankita. Someone else said, are these Jupyter notebooks open source and available to use? And I am proud to say from the DNA Nexus notebooks, often they are. So when DNA Nexus makes notebooks for sort of base stuff, we often put these on publicly available GitHub repos. And if you check out the DNA Nexus blog posts on Medium, often they'll point to those notebooks. Sometimes those notebooks sometimes they sit on GitHub. Other times they actually sit on the DNA Nexus platform. And all you need to do is go and get a DNA Nexus account. You can go to these public DNA Nexus. You can go to these public DNA Nexus projects. even if you're not a DNA Nexus user, and download them for your own use. In terms of, thank you, Brenton. And in terms of the notebooks that are built by MIODx, I will let Ankita answer that question.
Yes, so the MIODx product it's a paid subscription, so it's not an open source Jupyter lab. We have built applets that we've put out in certain communities for restricted use. However, it's not the Jupyter code. It's there's a lot of know-how that's gone into building the product, and it's not an open source. So that's,
right.
Yeah.
So what she's saying is, if you would like access to those notebooks, you should contact MIODx and see about working out an arrangement with them. All right. Here's another question. How did you get these tools created on DNA Nexus? Did you have to learn how to develop on the platform?
Yeah, I can answer to that. So the approach we took is a hybrid approach where, so DNA Nexus essentially has a team of scientists that will work with you on a service basis. And we scoped out the pipelines and did, based on our know-how and understanding of the biology and the need of the market. And DNA Nexus built some of it and we validated it. And as a second round of iteration, our internal bioinformaticians have also built tools with training from DNA Nexus. So this is, again, all happening on their platform. So you can run it, as Ben said, using Jupyter, but also build applets that are automated. So, yes.
Thanks, Ankita. I mean, it's really great that you are answering these questions. In lieu of me, I think that was way nicer of an answer that I would have given. So there's two related questions I see. So let me ask both of them together. I'm sure some details you'll be wanting to give and others maybe not. So the first question is, and these are from two independent folks, but the first question is, can you give more details of your PCR and library making? How does it have less bias? And do we have to use your kit to generate data? So that's one question. And then another question that I thought was related is, what is the sequencing coverage required in MIODx? I'm not quite sure what they mean by required, but I'll let you field those before we move on.
Yeah, so I think this is all going to, so there's actually a lot of questions in this. First of all, the kits. So our platform for just running analysis, not machine learning, is kit agnostic. We've validated various kits, and based on our experience and all the benchmarking studies, we've picked to recommend or to do our studies using the Takara kit, which is a 5' and a template switch oligo-based bulk TCR. The CDR3 question, I'm sorry, not the CDR3, but the sequencing depth is what is recommended. It's sample driven. So again, here we're using a lot of public data, so we had no control over it. However, we normalized it with our machine learning algorithm. But if you're going to generate your own data, if you're running like diverse samples such as PBMCs, you probably want to go the deepest, like, it's 1 million reads per sample. This is driven by input and it's very experiment specific. I can get into this in much greater detail offline. A lot of this is public data and they were done and used in benchmarking studies. So hopefully that answers the two questions.
Yeah, I think so. And I think that underlies a broader point, which is, I think sort of in the 80s and 90s, we thought about like science kind of like the Jetsons, where like there'd be like a push button and we could, have answers to how to do science. And I think, honestly, as things progress, that's becoming actually less and less true. And we really need a community where we pair people with expertise in different things. And so people are able to communicate with each other. And now I'll stop pontificating and ask the last question I see up here, which I think is it's an interesting question because it can be contextualized several ways, which is CDR3 the most common repertoire? I think they mean receptor data. And do you need all CDR1, CDR2, CDR3, or just CDR3 data for analysis? I'm going to guess, and Kita's going to tell you, depends on what question you're asking, but I'll let her answer this for herself.
Okay, I think Ben could have also answered this, but is CDR3 the most common eye receptor data? Well, it depends on, again, the way the eye receptor is set up. It's a public database, so whatever has been deposited. So if you look at, if you're selecting adaptive, yes, it's going to be only CDR3, but there is lots of full-length data available, meaning BDJ data. And as far as our analysis, we can do either way. We can just run CDR3 only mode, or so the point being, you don't need to have full-length chemistry to run the analysis. I think that's the question. So I think, do you need both T and, oh, sorry, Ben, I started reading the questions.
Oh, it's okay. Why don't you read the next question to me? Because I think maybe I'll take that one. So let's switch roles here. You go ahead.
Do you need both TCR, BCR, and phenotyping expression profiling as an example for machine learning?
No. So that's interesting about machine learning. Is that actually you can ask questions of any data set. But I'd like to give a bunch of caveats about machine learning. I mean, really, I think what we're seeing is that particularly in the biomedical space, throwing a whole bunch of data into a box and coming out with machine learning is actually not only unproductive, it's dangerous. So I think it's really important to think about your question very carefully and think about approaches others have taken to similar questions and try to responsibly answer those things. And I would also say that so often that that really I would say that so often machine learning becomes the beginning of an investigative process, not the end. So you find features, and then the idea is to chase them down, to validate, to do all those types of things. That said, you can do some really interesting machine learning with just phenotypic data. So if you think about how much physical activity is related to different cardiac pathologies, for example. I mean, that there are questions there that you could use machine learning for, if you had, say, Fitbit data, for example. So it's really not sort of which data type you have, but how much data. And actually, that it gets, I think the next question you're asking is sort of about the triad and thinking about matching between peptide and HLA and TCR. And I would say that, I mean, really, there's some people doing amazing work on that. And there are machine learning models you can apply. But what we don't have enough of for really sort of being able to look at the triad in a large scale way yet is harmonized data. And really, we need a lot more harmonized data. Also, a lot of folks have tried to use, over the last couple of decades, have tried to use structural biology approaches. And structural biology approaches are very frustrating because of glycosylation. And so that's something to think about too. So, I remind people that both in tech and science, there are a lot of smart people. So if there's a question that's very motivating, but nobody has made all that much headway yet, there's often a reason why. That said, I think it's a very exciting time because the amount of data, as well as the approaches, and also importantly, the operationalizability of these approaches. So being able to take these esoteric machine learning approaches and being able to put them into an operational Jupyter notebook where lots of data scientists can use them in a cloud framework that's big enough to do so, is now all coming online really fast and together. So I think over the next 5 years, it'll be a really exciting time to see a whole bunch of new developments.